can check out the percentage on things.
Okay.
Good.
All right, so my presentation is on developing some drug candidates for the MET inhibitors for the treatment of NSCLC, and those are my two mutants that I'm going to be targeting. So NSCLC is a lung cancer where there's an abnormal growth of cells in the tissues of the lung. It's categorized into 3 different types of lung cancer, and it's based on what type of cells it's targeting. For example, adenocarcinoma is the most common type of NSCLC, and that's where it affects the outer areas of the lung. The MET inhibitor is a tyrosine kinase receptor, and it's located on the chromosome 7. It's activated by hepatocyte growth factors, and it phosphorylates secondary messengers to cause the overexpression of cells and abnormal growth leading to cancer. The current drug on the market is tepotinib, and it's used for metastatic non-small cell lung cancer, and it inhibits the MET so that it doesn't phosphorylate these targets to cause the overexpression of these cells. So the goal of this project is to identify drug candidates to inhibit this receptor for the treatment of this lung cancer. And the goal is to really replace current treatments that are on the market. So the key scientific problems that are to be solved in this experiment is to screen for the top 5 compounds, identify IC50 values to compare to the current drug on the market, and try to find some new target populations because the current drug on the market only treats adults and not children or female that are pregnant. And we also want to improve some of the side effect profiles of the drug and also try to help diagnose the drug as the common risks or the common symptoms that are associated with this is like coughing and wheezing and things that are commonly associated with other types of diseases. This is the research approach, and highlighted in green is the computational portion, and highlighted in blue is the kinase inhibition assay or the wet lab portion of this lab. The wet lab is going to be repeated into 2 portions for the wild type and for the mutant to determine the IC50s to compare to the current drug in the market. And then for the computational portion, it's going to be similar to what we did in class. So to highlight the importance of computational chemistry in general, I really wanted to draw a comparison to COVID-19. So for typical drug discovery, we see it happening in about like 12 to 15 years, and it costs around $2 billion to discover a drug from hit to lead. So current methodologies that we can use with computational chemistry shortens this time, and any aspect of drug discovery that we can shorten is going to be crucial for getting to the market. And this is really important in terms of revenue for any company or when we discover these drugs, because the time that we get to the market is essentially dollars that the company can be making. So the research plan is to identify the top 5 compounds, and then we're going to measure the luminescence in the wet lab at 0.1% of the IC50, the IC50, and 10 times the IC50. We're going to use the same protocol that we use in the wet lab at the same concentrations and do 2 repeat experiments for the wild type and for the mutant. For future studies, we want to do in vitro and vivo studies, ADME studies, and possibly test other FDA drugs. One thing I wanted to highlight is that we use like 1 microliter of compound, which is a really small concentration that was hard to really check with the luminescence. So I was thinking of maybe trying to dilute these stock concentrations to see if that could help so we can put more concentration of the initial stocks into each IC50 concentration that we test. And these are the expected results of the study. Highlighted at the top is the current drug that we have, and these are the 5 compounds that were found by the bioinformatics students. Highlighted in green would be, these are fake results, that would be the best compound that could potentially be used for further studies. And also highlighted in the right is the graph that we created in lab, but hopefully like the results of this study, the concentrations will decrease over time. That is it.
Question.
Yes.
What is your time frame for all these experiments?
That's a good question. The computational part, like to the market, you mean, or like
Like to the end of your publication?
I think that's
I mean, anything that can cut the typical drug discovery time is the goal. So I'm saying under like 12 to 15 years.
cool.
Yeah.
Did they say what the reason was for like, for a new drug, like why your drug wasn't the best?
It's not targeting all patient populations, like it's only specifically for adults. Like, so if you could find something that maybe helps for children that have this disease, that'd be helpful, or even females that are pregnant, but those are two really hard populations to test for. So it's understandable why they couldn't really have results for those.
So part of how you want to change the experiment is you want to scale it up. Basically, how do you
Yeah.
plan to account for the cost of producing more drug in order to do these scale ups?
That's actually the exact reason I want to scale up, because like when I used to work with viruses, right, we get a very small concentration and that's very expensive. So like scaling up, I mean like taking that stock concentration and diluting it so that we can like use more. But it's not like actually like increasing the concentration, which is going to increase cost. So it's taking that same bottle and amplifying it so that you can use more than one microliter.
Okay, so I think it's a great idea about targeting some particular population, children, women. So then my question is that what kind of property you like to optimize? For the receptor, it's the same receptor. Then what kind of property will the nice to the children? Then what kind of tool we need to optimize?
That's a question because we don't, for like normal drugs, we can't really test these populations. Like females, like that would be teratogenic. I mean, it could have birth effects for the kid as well. So like,
So basically the tolerance would be low, right? Because they are a baby.
yeah.
And what about children? Children, the public organ has not, so they're not. So again, the tolerance would be low. Then what kind of property we need? Those population.
You mean like more specific for the receptor? Like maybe it's subtypes, things like that?
For example, children with chronic improvement, so what kind of property? The tolerance are low, so
Oh, like maybe also like a different dosage form, like because it's oral. So maybe for drugs.
So if drug more potent, then we need dosage.
Mhm.
Because that's the first feedback. And what about the
Of the drug.
drug? Now we go for those very sensitive population, like children, they are organized. Baby. So the tolerance is very low. That basically means that sensitivity for all those property we'll say less toxic. Then what kind of tool it will work the virtual screening control. We have the tool.
Or like making it more specific for that ligand.
It might be put up to reduce the toxicity, increase the reduce the side effects, those kind of things. So we can check the for this, we have the stock, right? So that's the area to optimize for other drug quality.
Oh,
Here is the bonding.
Yeah.
Here is potency of bonding. This is just one drug cost. The other topic we can optimize is the aluminum plus toxic. Right?
yeah.
So those products can be optimized with properly. Yeah, so yeah, there's two of them optimized.
Yeah, is that like what the Desmond? I think Annie was talking about it. I just wasn't sure how to use the program. That's why I didn't hear talking about it. Yeah. Okay.
Okay, good.
It's amazing.